onward to the Hershey and Chase experiment. Alright, so there are two experimental groups. We're going to start with the one that's pink, and then we'll go with the one that's blue. So in the first experiment, Hershey and Chase, both a man and a woman, grew viruses, that's what phages means, viruses, in radioactive sulfur. Now when you label something with radioactivity, it allows you to see it, okay? So this is what's supposedly here, this is the viruses growing in radioactive sulfur. And radioactive sulfur labels the protein, because only protein has sulfur. DNA does not. And then he let the viruses, or phages, infect the bacteria. Okay? Then he put this sample here, with the viruses infecting the bacteria, into a blender to allow the viruses to separate from the bacteria. Otherwise, they would just hang on there. Okay. So as you see, after running it into a blender, the viruses are now separated from the bacteria here. Okay. Then he ran the mixture in a centrifuge. And what the centrifuge does is it separates things by size. Okay? And bacteria are much, much, much bigger than viruses. So what happened in the centrifuge is all the bacteria ended up in the pellet, okay? And all the viruses here ended up in the supernatant, or the liquid. Okay? And what he found at the end of the experiment, if they could measure the radioactivity, which remember was labeling the protein in this case, because it was sulfur that was radioactive, what he found is that there was no radioactivity in the pellet. The radioactivity was only in the supernatant, where the empty virus shells are. So from that, because there was no radioactive sulfur found in the pellet, and remember radioactive sulfur labels the bacteria, he was able to conclude that it wasn't the bacteria that went into it wasn't the protein. It wasn't the protein that went into the bacteria, because the bacteria is in the pellet. The bacteria stayed outside the bacteria. So from that, he was able to conclude that it wasn't protein that was the genetic material. Okay. Now, in case 2, he grew the phages, or viruses, in radioactive phosphorus. And radioactive phosphorus labels the DNA, not the protein. And he did the same thing. He let the viruses infect the bacteria cells, and he ran it through a blender, and then he ran it through the centrifuge. This time, he found the radioactivity in the pellet where the bacteria were. Remember, in this case, the radioactivity was labeling the DNA only. So from this, he was able to conclude that it was the DNA that entered the bacteria, and therefore it was the DNA that was the genetic material. Because at that time they knew that viruses injected their genetic material into bacteria, they just didn't know what it was. Okay, so this summarizes the experiment. So when they labeled the virus phage is a virus, with radioactive sulfur, okay, they found the radioactivity in the supernatant. Okay? And they found out that the protein stayed in the supernatant, therefore, it wasn't the protein that infected the bacteria. And then when they labeled the phosphorus, which labels the DNA, they found that the DNA was in the pellet. Therefore, they concluded that it was the DNA that entered the bacteria, and therefore DNA is the transformation factor. Now I know this experiment is very confusing. Ralph seems to get it, but if you would like to watch the video explains it pretty well. I would suggest watching it. I've got one more experiment. This one's easy. So, in the last experiment, Chargaff took many samples of DNA from many organisms and analyzed the bases that were present. And what he found is that the number of A's was always roughly equal to the number of T's, and the number of G's was always equal to the number of C's. Now, Chargaff did this before Hershey and Chase. I'm sorry, not before Hershey and Chase. Before Watson and Crick discovered the structure of DNA. So they didn't know at this point that A pairs with T and G pairs with C. But they were beginning to believe that that might be possible, since the number of A's were always about equal to the number of T's, and the number of G's were always equal to the number of C's when they took DNA from any sample, from a human, from a cat, from a tomato. They found that the A's were roughly equal to the number of T's, and that the G's were roughly equal to the number of C's. And this information actually helped Watson and Crick, and also poor Rosaline, who got her data stolen, discover the structure of DNA. Alright, I think that's it. Yep. Alright, answer the guided questions. See you guys on Monday.